everybody. My name is Raul. I'm a senior software developer at the Sanger Institute. I work in the cancer division. And it's a pleasure to be here. It's a great source of pride for me. I lived in many years here in Barcelona. And it makes me very proud that this piece of software is being developed here. Plus, now I have like a great excuse to come by to enjoy this city. And today I'm going to talk to you about taking a sequencing protocol that we have been working at Sanger and putting it into a Nextflow. So this sequencing protocol is called Nanorate sequencing, and it has the unfortunate title a little bit because which usually shorted to NanoSeq, which can be very confusing because Oxford Nanopore also has something that is called NanoSeq. And it's basically borrows from the idea of duplex sequencing, where you have, during library preparation, you attach unique molecular identifiers to each fragment on both ends. And then you PCR amplify and sequence. And those unique molecular identifiers are then grouped together. And that allows you to get rid of PCR artifacts and the sequencing errors. So it allows you to tell with very high precision presence of somatic mutations. They estimate about 5 errors per gigabase of sequence. And this technique has been worked at the lab of Martincorena. And here I have the official repository of the code. And basically, the developers, myself and Federico Abascal. And when he handed over the code to me, so he works both lab and does the computer stuff. So I sat with him, clean up the code, because, I mean, his code is kind of messy, so I had to go through it and take out all the hard-coded references and all of that stuff and put it into a repository and apply good practices to the software. And another thing, it's very diverse set of programs, so ranging from C++, C, R, Python, Perl. So the first thing that I did is write a wrapper for all of these, so the analysis would be simplified. So you can see it's run by a series of subcommands that are on the right side. So basically, you do a quick coverage calculation to estimate how many reads you have in each area of the genome, then partition the genome according to this, and calculate tables that are spread over many jobs. And then using those tables, you compute the variance, SNVs, and indels, and finally put everything back together into just a BCF file. So I have been working on this code for several years now, since almost joining Sanger. And about 8 months ago, there was a Nextflow training course at the EBI. And I attended. It was like a three-day session. So I thought it would be nice to put this into an Nextflow. Everything had been driven by LSF arrays and Bash, basically, which made it a bit hard to rerun and to manage the analysis. So this is like my first attempt for the workflow. Basically, I have this is a paired analysis, so I have like what would be a tumor and a normal BAM going into the analysis, and the steps that I outlined previously. And of course, I wrote it in the ESL1 because it seemed like the simplest thing at the moment. And so I took this and went to the scientists. I was all excited, and they said, of course, like, hey, could you add more stuff to it? Because so yes, they actually told me, like, we spent a lot of time in the pre-processing of the BAMs. Could you add that to the workflow? Yeah, of course. And so now I have steps where I could remap the experiment, because maybe they wanted to change the reference, say, from GRC 37 to 38, and then add do the tag processing that is required for the experiment, plus also a couple of quality control steps in the workflow. And at the end, also added some other post-processing calculation. I also migrated to DSL2, which, after having done that, I tell people in our group that they should probably directly start with DSL2, because I found it not so straightforward to migrate my pipeline from one to the other one. And I also stumbled upon NFCore, and although I didn't take the entire template, I liked some of the ideas and incorporated it into this pipeline. Especially, I liked the metamap. I thought it was a very good idea, so I put the metamap into this. So again, I went and talked to the scientists and asked them, what do you think? And again, they said, could you add more stuff to it? So now they wanted, at Sanger, we keep our files in an iRODS storage, and scientists usually don't like to deal with that, which I understand because it's command line driven. And so they said, could you write something in the Nextflow so that retrieves the files for us? And so you just specify a project number and the samples, and then the pipeline goes and gets all of that for you. And here, I also started struggling with something that I think many people probably have encountered. If I should limit my analysis to one Nextflow per analysis, or do a batch of analysis with what Nextflow instances. And I think either approach has its benefits. I, of course, chose the more complicated one because I thought it would be, I didn't want to take too many resources with Nextflow instances running on our cluster. So I decided to make batch processing available in this version in the third version of the pipeline, which complicates a lot the handling of the channels. So in here, I really thank the people in the Nextflow Slack, because I was really lost for several days. But I was fortunate enough that the Slack channel probably had just started, so people were very responsive. And they helped me out to figure out, because like the first part that you see, all the download cram, all the way to the coverage is done per sample. But then you have the complication that then the calculation gets spread over hundreds of jobs. So then you have to do clever merging of the channels. Plus, now I also added everything, turned all the processes to use CRAM, which was also kind of a very fun experience. And the workflow there, I'm pretty happy with it now. It's working. I think now the problems that we have are mostly in the software itself. But now I started working in another project and really pushing the limit. In here, we are doing cohort analysis of 20 or to 30 samples. That is just one analysis. So it's like here I have one of the reports, the first report that I was able to generate with that pipeline. So 12,000 processes for one analysis. And this is basically doing SNBs and indels on cohorts of samples. And we have to run hundreds of this type of analysis for this project. So really, Nextflow is opening this project, because we couldn't do it in any other way, because it would be very difficult to manage all this analysis in any other way. And this brings me also to something that I find we have an issue there at CASM, at the cancer division at Sanger. So we have a very nice graphical interface where researchers typically launch their analysis. So we have all the core algorithms in there, and they just click, which of course they like very much, because we have clinicians and people who are not very computer savvy. And but this is also very restricted. because I've had to port algorithms into that pipeline, and it's very painful. It's very monolithic software. So I think I was very excited to see the talk about junction, because I think we having a parallel thing where we keep our pipeline very monolithic, which is a very good cookie factory, but also having flexibility, so researchers could design their workflows without being tied up by the way that the pipeline that we have now works. And with that, I just want to thank Federico, who has co-developed the code with me, and Alex Kagan and Matt Neville have been kind enough to try this on their projects. They have projects that are using NanoSeq, so they are the ones I have been iterating the software. And I want to thank very much Phil and Federica, because they weren't, actually I had given up on that idea of doing the batch processing at some point, because it took a couple of days, but they did come back to me, and so I was very happy with that. I was almost going to give up on that. And with that, I think that's pretty much all I have to say today. Thank you.
Great, thanks so much. It's 15,000 CPU hours, is that right, for the pipeline?
Yes.
Decent, very decent. It's got a question around how long did it take you to port the pipeline into DSL2?
So I understand from DSL1 to DSL2. It wasn't that bad, because I already had kind of the experience. The processes were simple enough, but the channels, I did have to rework the logic a little bit, because the logic of the channels changed, no? So yeah, it took a couple of days, I would say, to refactor.
And what was the lowest VAF, I understand this is variant allele frequency, that you're able to detect with the UMI-based protocol?
I'm not really sure. I'm not a specialist in that, sorry.
Yes. And there's another question from Maxime. Is there any plans to collaborate or try and include or join these efforts with NF Core?
I've struggled with that question, and I asked other members, other groups at Sanger about this. And the thing is, I put a lot of optimizations in the code for our environments. So I find it a little bit of a compromise between writing something very general and then having like a good performance in our cluster.
And then how was the performance? Originally, I understand this pipeline was an LSF. We're using array jobs or array jobs. How have you found that performance with Nextflow, which, for example, doesn't support those kind of array jobs like that?
Actually, I like it better that the arrays, in my mind, it just makes it easier to punch group calculations, no? So even the managing of calculations is done well with Nextflow, because, for example, if I had to kill an analysis, I just kill the Nextflow instance, and that takes care of and kills all the related jobs. So it makes life
cool.
easier.
Awesome. Thanks a lot.
Thank you.